Okay. Good morning, everyone. My name is Ben Klein. I'm a current PhD candidate at Michigan State University, and I'm excited to be sharing some of the work that our lab has been doing on coastal brook trout along the Minnesota shoreline of Lake Superior. And Eric and Chris and Corey did a great job setting this talk up, so I'm looking forward to sharing our findings as well. So as we've already talked about today, brook trout are one of two native char in the Lake Superior Basin, and they're an iconic cold water species for their ecological, economic, and cultural value to the region. However, despite their innate value, we've observed declines in both phenotypic and genetic diversity in brook trout in the Lake Superior Basin over the last 100 years. And as Corey mentioned, a lot of this may be attributed to anthropogenic activities such as land use change, wildfire, mining. But in this study in particular, we're also interested in the influence of the presence of these large natural waterfall barriers present in many of the rivers tributary to the lake. And as we've talked about already this morning, in the Lake Superior Basin, we see two unique life history forms of brook trout, the resident, which spend most of their life cycle in the streams and lakes tributary to the lake, and then coastal brook trout, which we define as any fish that's found in the lake proper, any fish that uses the lake for a portion of its life cycle. And so, of course, it'd be remiss not to mention the fish community objective for brook trout, which is to maintain widely distributed, self-sustaining populations in as many historic habitats as practical. However, there are a few key challenges to being able to do this. One is assessing our current knowledge of the relationships between coaster and resident brook trout. So, for example, can we think about where coasters are being produced, and also how the presence of these large barriers to movement influence these relationships. And additionally, we're interested in understanding how we can best prioritize management activities to conserve these populations for generations to come. For example, thinking about the efficacy of stocking, as well as the genetic health of the populations that we find along the shoreline. And so we used a genomic approach to be able to start to tease apart some of these challenges. And we used a genomic data set based on the single nucleotide polymorphism, which Chris mentioned a little bit earlier. So these are genomic data, and really what they are just single base pair differences across the genome. And so just for an easy example, fish in the blue population on the slide might have an A at a specific region of the genome, whereas fish from a green population might have a T. And so really what we're trying to do is quantify all of these small differences across the entire genome to be able to resolve populations and answer the questions that I talked about. We used an approach called restriction site-associated DNA sequencing, which really all you need to know is that this is a way to rapidly and cost-effectively do this in a lot of individuals. And so we're able to survey thousands of regions of the genome across thousands of fish across the basin to really get good power to be able to answer these questions. And so our final SNP data set was comprised of fish from 24 rivers tributary to Lake Superior along the North Shore Minnesota shoreline. Individuals sampled within the rivers were comprised of populations sampled both above those large natural waterfall barriers as well as individuals sampled below barrier. We also had 4 sampling locations within the Lake Superior proper, and these surveys were in line with the ones that Corey talked about in his previous talk. But all the fish sampled in this study were sampled approximately between the years of 2018 and 2022. And a big thanks to the folks at the Minnesota DNR for the sample collection, and especially the folks from the Coaster Genetics Project who contributed greatly to all of the angler samples that were included in our analyses. In total, we had about 1,600 wild brook trout, and all of those individuals were genotyped to about 8,000 SNPs, which were those genetic regions that I talked about on the previous slide. I also wanted to mention that we did include some domestic brook trout from hatchery sources, known hatchery sources that were used in some of our downstream analyses, which I'll talk about later. So getting into some of our findings, we used a multivariate approach known as the discriminant analysis of principal components to assign individuals to genetic populations. It's very difficult to show data for 1,600 fish in one plot, so don't worry, I'll translate this to be a little bit easier to read in a slide or two here. But here, each bar represents a single fish, and the value on the y-axis corresponds to the assignment to a given genetic group. And so here, the colors represent the genetic population to which a fish was assigned, and the groups of bars on this plot are organized by waterway from which those fish were sampled. And so the clusters of bars are arranged from north to south along the shoreline, so cluster 1 being the northmost stream, and down at 26 and 27 being the southernmost stream of the waterway. And the last thing you need to note here is that the numbers correspond to the geographic sampling location, and if there's an A or a B, that corresponds to whether that fish was sampled above a barrier or below a barrier. But that was a lot. I'm just going to walk through what you need to know. First is that using this approach, we were able to assign over 90% of our individuals to a genetic population, which was really exciting. And our first main takeaway is that we see a lot of population structure by the geographic sampling location, particularly within the same stream. So for example, if you look at this cluster here, at Kimball Creek 4A and B, those are fish that were sampled above barrier in Kimball Creek and below barrier in Kimball Creek, and we see that they look really genetically similar. In fact, even more genetically similar than fish in the neighboring tributaries. So to make all that data a little bit easier to digest, I've just taken everything from that plot and summarized it into pie charts on this map. And so here each pie represents one of the sampling locations, so all of these being our above barrier, right along the shoreline being our below barrier, and then we have 4 locations within the lake proper. And so again, the colors here correspond to the genetic population assignment for individuals within a sampling location, and then the proportion of individuals that assign to each of those genetic populations is depicted by their slice of the pie. And so just another way to reiterate that when we look at our above and below barrier populations within the same stream, they look really genetically similar. If I'll draw your attention to a second to the coaster sampling locations, so these 4 that kind of jut out into the middle of the lake, you might notice that there's a lot of colors contributing to these coaster populations. And that tells us that there are multiple within river source populations for coaster brook trout. To make this a little bit easier to think about, I've drawn arrows for populations where greater than 10% of the population is from an alternate source from where they were sampled. So, for example, if we look at this orange population here, this is the Poplar River, we see that fish that were assigned genetically to the Poplar River actually were sampled in 3 of our 4 coaster sampling locations. So fish are moving a lot across the lake, and we're seeing that there are multiple sources of coaster brook trout along the Minnesota shoreline. I also wanted to point out that all of these fish that are moving in the below barrier reaches are exchanging, and these below barrier movements portend access to lake habitat. So by our definition, all of these fish that move from one below barrier reach to another would be considered a coaster brook trout. But what about the other individuals I mentioned that didn't assign to a wild population using the approach that I mentioned before? So I said that about 92% of our individuals we were able to assign to a wild source population. What about the other 8% that were maybe a little bit more ambiguous? So I used a type of formalized genetic assignment testing using our known genetic source populations as a reference. And this time I included all of our known hatchery strains as a reference as well. And we created assignments for those individuals that were unknown using the first approach. And so I want to draw your attention to this big block here around Grand Marais. And these are all fish that we'd consider coasters. They're sampled in the lake proper. And you might see that there's a big blue chunk. So what's going on with this? So one possibility is that some of the coasters that we're sampling in Grand Marais are actually stocked hatchery fish that have been recaptured in the wild. However, it's a little bit more nuanced than that. If we think about our hatchery broodstock sources, it might be very difficult to be able to distinguish hatchery broodstock, for example, sourced in the Nipigon region, from a wild Nipigon fish that simply moved down into the Grand Marais area. And so with the genomic approach that we've used, it's really difficult to be able to disentangle those sources. A third possibility, though this seems less likely, is that these coasters could be from an unsampled wild source population, but as Nadia shares a little bit more about some of the research that we've been doing in other parts of the basin, we're starting to get a more comprehensive look across the basin and have a good idea of where our gaps might be. Finally, I just wanted to show a little bit about where our coasters are coming from, spatially across the Minnesota shoreline. So here's just a simple heat map where the darker colors mean that we have more coasters from a given region. You can see that the midshore region appears to be a major source for coasters along the shoreline, though I did want to point out that most of our samples are from the midshore area, so this could just be an effective sampling. And our final goal was to be able to infer the genetic health of the populations that we resolved in this study. And so there were 15 genetically distinct populations that I've showed based on the colors on the previous slide. And we estimated effective population size, or NE, for each of these 15 populations. And so NE is an evolutionary analog to census size. It's a really powerful monitoring tool, and we can use it as a proxy for understanding genetic health of a wild population. And generally, when we think about NE, we want to try to manage populations to stay above critical thresholds, which I've animated onto the plot here, but generally, for a species to remain stable, we want to manage NE to be greater than 50 or 100 in most cases. So here's our NE estimates for the 15 genetically distinct brook trout populations that we identified in this study. I've animated on the critical values of 150 to make it a little bit easier to see. And our main takeaway here is that effective population size is above the critical threshold for most of the populations sampled within our study, which bodes well for brook trout in this region. I did want to point out that for the two populations that dip below the critical threshold of 50, we did have relatively low sample sizes of 18 and 38 respectively, so those NE estimates might be a bit more narrow with additional sampling. And so going into this work, I just wanted to reiterate that some of our main challenges were that we had a limited knowledge of the genetic relationships between resident and coaster brook trout, and we wanted to gain a better understanding of how our management activities influence the conservation of brook trout in the basin. For questions 1 and 2, we determined that populations from multiple rivers act as sources for coaster brook trout, and this information aligns very well with what Chris shared earlier with some of the work that they've done in Ontario. We wanted to note that coasters may be stock domestics, but it could be likely that they're also wild migrants from hatchery broodstock sources, or even individuals from unsampled wild populations. And finally, in terms of the genetic health, we noted that NE estimates are above critical thresholds for survival for most populations along the Minnesota shoreline. So tying everything together, I just wanted to reiterate that brook trout along the Minnesota shoreline of Lake Superior harbor a lot of unique genetic diversity, and this was reflected in the large degree of genetic population structure that we observed in this study. Management decisions focused on preserving this unique genetic variation found in these populations will be critical for sustaining brook trout in the Lake Superior Basin, and Chris spoke really well to this earlier, but I just wanted to reiterate that stocking approaches that preserve and maintain the unique genetic diversity found within the region will be critical, and we're just beginning to uncover some of these adaptive differences and these local population level variation, and we're poised to gain a lot more insight into that with these data sets. And finally, I just wanted to reiterate that it's really important to consider individuals in these below barrier reaches in our conversations around coasters. We found that there is a lot more below barrier movement than maybe we might have previously anticipated. And so when we think about coasters and their conservation, it's important to consider this below barrier connectivity as well. So I wanted to thank everyone who contributed to this project, especially the folks at the Minnesota DNR for all of their efforts in sample collection. And if there's time, I'll be happy to take any questions.